Hello and welcome. Today, I would like to invite you on a journey to explore DNA not just as a static code, but as something far more dynamic, a fractal antenna. As an antenna, DNA requires a signal source, a field that carries morphogenetic information, collective memory, and even elements of group consciousness. And so I ask, what if we are not just passive, helpless victims of our genes? What if illness is not the result of a broken code, but the echo of unresolved information carried in our personal and ancestral field? Let's go deeper. What if DNA does not just carry biochemical instructions? What if it also functions as a receiver for patterns encoded in our surrounding informational field? Scientific research increasingly shows that DNA is highly sensitive to resonance, structure, and environmental signals. We also know that trauma, whether ancestral, emotional, or environmental, can leave long-lasting imprints. These imprints may not just be in the body, but around it, in a kind of field memory. DNA in this model becomes the translator of this field. It is not only a code, but a conformation, a shape that shifts depending on the signal it receives. In this light, illness becomes less of a genetic failure and more of a misalignment in the field. In modern medicine, we often use tools like sudden blot or DNA sequencing to identify mutations or genetic predispositions. But what are we truly seeing in these maps? Are we only reading a code, or are we witnessing a snapshot of the field's influence on that code? Patterns of trauma, suppression, loss, or fear may not just live in our psychology. They may subtly shape the very structure of our DNA. So when we talk about a genetic predisposition to cancer, depression, or autoimmune disease, perhaps what we are really seeing is the visible footprint of an invisible signal, a field echo that has crystallized into biology. In this view, genetic mapping becomes more than a diagnostic tool. It becomes a window into the soul's unfinished business. So what happens when we intervene directly in the genome? With technologies like CRISPR, we can now cut, remove, or insert genetic material with remarkable precision. Or so it seems to be like that. After such intervention, scientists often return to tools like sudden blot to verify that the error has been corrected or not. But here is the question: who controls what is considered an error? And what counts as a correction? Even the most advanced gene editing tools can have off-target effects, altering areas of the genome we never intended to touch. More importantly, what happens to the signal? If DNA is a receiver, not just a blueprint, are we correcting a pattern, or simply overwriting a message we don't yet understand? And what are we doing about the long-term consequences of editing a body without addressing the field that shaped it? Now let's imagine a different approach. Instead of editing the code, what if we could retune the signal? Plants carry more than just chemical compounds. They also hold vibrational profiles shaped by their form, their habitat, and their evolutionary history. A carefully selected herbal formula can act like a tuning fork for the body's field, helping to harmonize chaotic patterns and restore coherence. For example, a combination like reishi, danshen, and baihe may not only affect the cardiovascular or respiratory systems, but also resonate with deeper aspects of the lung, heart, spirit axis, often impacted by grief, exhaustion, or trauma. This is not just chemistry, it is biofield orchestration. Instead of forcing a change, we invite the system to remember its original song. As we explore resonance-based alternatives to gene therapy, we also need tools to stabilize and integrate the shift. Eclitoprostrata offers DNA protection during field transitions. Its antioxidant profile helps preserve structural coherence in the DNA water matrix. Ligustricum lucidum, on the other hand, nourishes the deeper tissue water shell field, anchoring the cellular identity and supporting post-intervention recovery. Together, they create a botanical buffer, protecting both the genetic material and the energetic signature of the person, allowing new information to be received and held with clarity and resilience. When we work at the level of the field, something profound happens. The DNA does not need to be cut or rewritten. It begins to soften, to relax its defensive conformation. Why? Because the distorted signal that once shaped it is no longer present. The body's structured water matrix, which surrounds and interpenetrates every cell, responds to coherent frequency, especially from plants, sound, breath, or emotion. As we harmonize the field, cellular communication improves, energy flows more freely, and epigenetic expression shifts naturally. In this model, herbs don't impose, they remind. We don't override the pattern. We clear the static so the original design can emerge. For this approach to be credible, especially in a medical or scientific context, we need both subtle intelligence and solid measurement. That's where sudden blot comes in again. We can still use it to track changes in DNA structure or methylation patterns. This reassures the evidence-based mind. Precision is important. Even if we harmonize the field and support the DNA with plant-based resonance, there is still one more crucial element: conscious integration. The patterns that shape our health often carry deep emotional roots. Grief, shame, resentment, and fear. For example, a genetic predisposition to colon cancer may reflect modern biology. It could mirror a long-standing inability to let go, to release what no longer serves us. Without addressing the psychological and behavioral layers, these patterns may resurface, even after the field has shifted. That's why this approach calls for active participation. Breathwork, somatic release, guided introspection, whatever helps the psyche complete the loop. Healing is not just correction. It is a process of reconnection. As the heart of this conversation lies a fundamental choice. What kind of intervention do we believe in, and why? Gene therapy represents a path of external control. It seeks to fix what has broken by force through edits, cuts, and engineered corrections. Often, it is driven by fear, urgency, and the desire to dominate nature. In contrast, plant medicine follows the path of inner coherence. It listens, responds, and harmonizes. It works with the body's intelligence, not against it. The difference lies not only in the method, but in the intention behind it. One path operates from separation and correction, the other from trust, alignment, and integration. The outcomes may look similar on paper, but the felt experience and long-term stability are worlds apart. In the end, perhaps the most important shift is in how we perceive DNA itself. Not as a rigid inheritance or a fixed sentence, but as a living interface between the inner and outer world. When we stop cutting and start tuning, when we shift from manipulation to collaboration, healing becomes less about fighting the disease and more about restoring harmony. This is not just an alternative to gene therapy. It is an innovation to listen differently, to work with the field, and to let nature be the guide.